Hi everyone, this is Mr. Sinti, and this lesson is on C3, C4 plants, kind of a problem that plants experience called photorespiration, and also CAM plants. So let's begin with a little review here of the Calvin cycle. As you can see, the Calvin cycle is very important. It's also called the light-independent reaction, and it's also referred to as the Calvin cycle. And let's get right into that. And so you could see, as you recall, carbon dioxide comes into the plant through the stomata. And when carbon dioxide comes into the plant, it simply diffuses into the chloroplast. And then in the stroma of the chloroplast, it reacts with this enzyme right here. And let me see if I can illustrate that in red. This enzyme right here called, this enzyme called rubisco. Let me go back there. Sorry about that. This enzyme called rubisco is sitting here in the stroma. It stands for ribulose bisphosphate carboxylase, and it reacts with ribulose bisphosphate, this 5-carbon sugar that's present in the stroma, and it attaches carbon dioxide to it, making a 6-carbon molecule, which then is quickly broken down into phosphoglycerate. You can consider one of them, or you can consider 6 of them if you consider 3 carbon dioxides coming in at a time. This process, carbon fixation, is the very first phase of the carbon cycle, and it's where carbon is inputted. And so, as you're going to see, there's 6 phosphoglycerates, and with the help of ATP and NADPH from the light reaction, these molecules are reduced ultimately to the end product of photosynthesis, which is glyceraldehyde phosphate. It's a 3-carbon molecule, and there's 6 of them. You may recall that 5 of those continue on in the Calvin cycle to regenerate rubisco. Not rubisco, but ribulose bisphosphate. And so those carbons are accounted for. But as you can see, these 3 that come in, you could siphon out over here 1 molecule of the 6 glyceraldehyde phosphates. And if the cycle goes around twice, you can produce 2 of these, and then ultimately the 6-carbon hexose sugar glucose. And then if you continue with this, you can make sucrose, or again, more and more you can make starch. And so this is how the plant brings in carbon dioxide. It reacts with this enzyme. It fixes the carbon to an already present sugar, and then it recycles it back, but then it is able to remove these little 3-carbon fragments. So it's rather clever and very important. Let's consider the next situation at hand. Plants struggle, obviously they struggle in warm climate, especially when it's very dry. And so what you're seeing here in this picture is this is the lower epidermis of a plant, and these particular cells that are highlighted here in sort of blue-green are guard cells, which are flanking the side of this opening called the stomata. And what plants are able to do is they're able to open and close their stomata. And what's interesting is when it's warm, they face the problem of dehydration, especially in an arid environment. And so in order to solve this, they can close their stomata from time to time. And let me see if I can jump out of this and get to show you this little animation over here if I can. You remember seeing this in class. If I were to click on this, you can see how... Oops, let me turn off the volume there. You can see out of the plant cell, lots of oxygen is being produced in the light reaction of photosynthesis. And the oxygen simply diffuses out of each of the plant cells, and it's in pretty high concentration in the spongy mesophyll, which is the lower part of the mesophyll in a typical plant leaf. What happens is that CO2 simply, let me let this play, the oxygen simply diffuses out of the stomata through those guard cells, and this is how oxygen appears in the atmosphere. Now, not all of it leaves the plant. Obviously, some of the oxygen is used by the plant itself in the mitochondria for cell respiration. But can you imagine for a moment if you were to close the stomata? Let me sort of back that up a little bit. Do you see how, let me let that play. Do you see how if you were to close the stomata right now, And why would you do that? Because it's very hot out. And why would you do that? Because you wanted to reduce water loss through transpiration. If it's blazing hot, obviously heat's going to generate a lot of water loss, and so the plant's going to die through dehydration. But here's the issue. If it closes the stomata right now, this concentration of oxygen becomes dangerously high for the plant. I just wanted to sort of show that as a visual. Let's go back to the slides if we may, if I can. The question really is. Let's go back to that, and let's see, let's go here. And so, okay, that's great. And so, do you see how these stomata are able to regulate water loss by opening and closing? This is my point that I'm trying to make here. And so, in so doing, on a hot day, they're able to conserve water. But here's the issue. If the guard cells close, the oxygen builds up rather dangerously. It sounds ominous, but it actually can be, because a problem occurs in the Calvin cycle when concentration of oxygen is too high. Let's take a look at that. So what we're talking about once and for all is down here in the lower mesophyll, called the spongy mesophyll. The oxygen levels are building up. And why are the oxygen levels building up? Because the stomata are closing. So if the stomata closes, oxygen's building up in this airspace down in here. Here's maybe a better shot here. So oxygen's building up, building up. Sorry about that. We have a software update right in the middle of our presentation. These are the kinds of things that can happen. So O2 builds up down here when the guard cells are closed. And so what's the problem? Let's take a look. The problem that we have is something called photorespiration. And it's a little tricky, but let me sort of just walk you through it a little bit. Just to give you a background perspective here, what you're dealing with is, this is the whole plant cell. This is a close-up of the chloroplast. Here's a mitochondria over here in the lower right. And here's an organelle, if you recall, called the peroxisome. And here's the Calvin cycle, which is actually occurring in the stroma. Here's our sugar, 5-carbon sugar. What normally occurs is carbon dioxide reacts to the enzyme ribisco, and it fixes carbon to ribulose bisphosphate, and it produces, if you recall, 6 phosphoglycerates. What can happen when, let me sort of highlight these things, let me make it green. What happens when oxygen is in high concentration? Let's sort of go like that. And again, why is that? Because the guard cells are closed. What can happen is the enzyme ribisco, which is sitting over here, which normally fixes carbon. Let's see, ribisco, like this. Actually, the real name of ribisco is not just ribulose bisphosphate carboxylase, it's also oxygenase. And what's interesting about that is that it has an affinity for oxygen as well as carbon dioxide. And these are somewhat competitive. And as it turns out, the substrate that tends to win out in high concentration of oxygen is in fact oxygen. And so if you can imagine and sort of think about this, if this enzyme is preferring oxygen over carbon dioxide, then there's no input of oxygen. Then there's no input of carbon. And if there's no input of carbon, then what you're going to get in the Calvin cycle is nothing more than this ribulose bisphosphate, which can then be broken down into phosphoglycerate, and then some glyceraldehyde phosphate can be produced, sure, but ultimately you're going to run out of that substrate, because without a constant input of carbon dioxide, how will you get the extra carbon that you need to produce glucose? So this is somewhat dangerous for the plant, because if oxygen were to come in, what it's going to do to that ribulose bisphosphate, that 5-carbon sugar, is basically fracture it. It's going to produce, let's look at just one at a time, it's going to produce one 3-carbon phosphoglycerate, which could continue on. But then it produces this 2-carbon molecule called glycolate. And I know the name's a little weird, but glycolate is this 2-carbon fragment, and some botanists believe that it's a waste product, because what happens is that 2-carbon is removed from the chloroplast completely. It actually leaves the chloroplast and it goes into the peroxisome. there's a little pathway illustrating the details of it, but the biochemistry pathway of this is an interesting one, but it's sort of not above us, but it's beyond the scope of the AP. And so what ultimately, this is called photorespiration, because ultimately that 2-carbon fragment gets sort of shuttled into the mitochondria right over here. And so this is sort of where cell respiration, if you recall, occurs. Ultimately, the bottom line is when oxygen outcompetes carbon dioxide because it's in high concentration, the production of sugar decreases, and therefore there's no glucose being produced, and ultimately it's a negative process for the plant. And so it's kind of curious on how something like that can take place after all these years, and I'm talking millions and billions of years of evolution. It's very curious on why Rubisco has a preferential attraction for oxygen over carbon dioxide. Some speculate, if you recall this in class, some speculate that perhaps it's a remnant of the past when oxygen was never really in this high concentration. Then again, some can speculate that it's a way to reduce the high level of oxygen in the cell, and because oxygen at such high concentrations can have deleterious effects on other biochemical pathways. So I can erase that. So what's the solution to this? Obviously, some plants thrive in arid, dry, warm climate. And a classic example of that is like the sugarcane. It seems to do really well. Some plants have evolved an alternative mode to carbon fixation, which minimizes photorespiration. It's like, how can they do that? Because Rubisco, they're not going to change their Rubisco. And so what is it? What is it about their leaf anatomy? What is it about their biochemistry that allows them to thrive in this environment? And the answer to that is this a plant anatomy called C4 and a biochemistry called C4. And so let me just walk you through this. If you notice here, the upper epidermis, over here is the lower epidermis. Here's the guard cells guarding the opening of the stoma, the opening where CO2 and oxygen come out. Do you remember that the cells, let me draw this in, the cells in the middle of the leaf are referred to as mesophyll. I don't want that to happen. Mesophyll cells. And what's interesting, in a typical C3 structure, the mesophyll cells are arranged such that in the upper part they're palisade-like and on the bottom they're mesophyll. But in this case, the mesophyll cells surround each vein. And what's happening in the vein, if you recall, the cells that surround the vein completely are referred to as bundle sheath. Here they're highlighted in dark green, right here, the bundle sheath. And so the mesophyll cells, which are out here, are going to be in light green. And then inside the purple is a special type of tissue called vascular tissue, xylem and phloem, that conduct the sugar and water through the plant. So what's interesting is, let me sort of draw this over here and highlight it in blue. So picture this, over here in the air spaces of a C4 plant, this is the C4 plant, over here in the air spaces you've got oxygen right in here, and you have carbon dioxide. And so the issue is that Rubisco has a preferential selection for oxygen. So what can a plant like this do? And so as it turns out, what the plant does is when it closes its stomata and oxygen's up really high, do you see over in these light green mesophyll cells right here, the mesophyll cells? They have a unique enzyme, and this unique enzyme is called PEP carboxylase. Let me just draw it in like this, PEP carboxylase. And so when it has a choice between oxygen and carbon dioxide, it chooses carbon dioxide over oxygen. And so when it does that, it takes carbon dioxide in like this, it attaches it in the mesophyll cells, so this is where we are, the light green, it attaches it to PEP. And when it does that, PEP is a 3-carbon molecule. When it attaches it, it produces this 4-carbon molecule called OAA. You may recall it from the Krebs cycle. It's the original substrate in the Krebs cycle. So a carbon is attached to 3 carbons, 1, 2, 3, and it produces a 4-carbon molecule called OAA, 1, 2, 3, 4. And this occurs in the mesophyll cell. So carbon fixation in a C4 plant occurs in mesophyll cells. Why? Because this way you can sort of keep oxygen out of the picture. You can just bring CO2, you're sort of sneaking it in. And so this is why it's called a C4 plant. You got this OAA, and then a little bit of a detail, but it's converted into malate or malic acid in the mesophyll cell. So you go OAA to malate. Then that moves through the plasma desmata into the bundle sheath. Now this is important. Do you see here in the dark green, the bundle sheath cells? This is where the Calvin cycle is going to occur. You're like, why is that? Because when malate goes into the bundle sheath cells, the carbon dioxide is like fumbled. It's released from malate. So there it is. There's the carbon dioxide. And then pyruvate is produced. Oh, we know pyruvate as being the end product of glycolysis. It's a 3-carbon molecule, as you recall. Let me just follow up pyruvic acid for a second. Pyruvic acid then comes back out of the bundle sheet where ATP attaches to it to regenerate PEP. So you got like a little cycle happening here. You got PEP, OAA, malate, pyruvate, PEP. It's a little cycle right here. But what you're doing is you're not really sneaking, but let's just say that anyway. You're sneaking CO2 into the bundle sheath. Oh, so CO2 is in high concentration in the bundle sheath. And yes, who's waiting for it in the bundle sheath, which is our good friend rubisco. And so if rubisco is here in the bundle sheath cells, it's going to be able to grab carbon and ultimately produce the 3-carbon sugar glyceraldehyde phosphate without any worry about oxygen being in high concentration. Because if carbon fixation occurs in the bundle sheath cells, we can keep oxygen sort of buffered, keep it back. Because over here the mesophyll cells are like, okay, we only want carbon dioxide to come in. And so oxygen is kept back. It's rather clever. And then obviously when sugar is produced, it goes into the phloem and then it gets carried on to different parts of the plant. So I find that to be one of the cooler things in the study of photosynthesis. And so photorespiration is the problem. And if it wasn't for this sneaking CO2 in, rubisco would be diminished. And so ultimately, the anatomy of a C4 plant and this unique biochemical pathway solves the problem of photorespiration. And so C4 plants this is sort of in words, C4 plant fixes CO2 first. And as you recall, it attaches it to PEP to produce OAA, and then into malate, and then ultimately into pyruvic acid, and then back into PEP. And so in the mesophyll cells, that's where carbon is being fixed. And in order to do that, there's a unique enzyme that prefers it. And that unique enzyme is called, it's a mouthful, so I'm hesitating. It's phosphophenyl pyruvate carboxylase, or PEP carboxylase. Let's go PEP carboxylase right there. And so this is the enzyme that if you were, here it is, it has a higher affinity for CO2 over oxygen. And so here it is. You got a choice between oxygen and CO2. What are you going to pick? What are you going to pick? It's going to predict carbon dioxide. And so this is where it attaches CO2 to PEP to produce OAA. And this occurs in the mesophyll cells. And so some examples of this is corn is a great example of C4 plants. We also looked at sugarcane in a second. So in the bundle sheath, that's where the CO2 is stripped from malic acid to produce pyruvate, and then ultimately CO2 is used by rubisco to start the Calvin cycle when it's in high concentration. And so as you can see here, the mesophyll is not the main site of carbon fixation, but rather the bundle sheath. And so if you take a look at this picture, I find this to be really cool right here. This is a cross-section of a C4 plant. And so it's like, what's the story? All of these cells are the mesophyll cells. And so out here, let's say that this is an airspace. And so you got oxygen here, and you got carbon dioxide here. And inside the mesophyll cell, this is where PEP carboxylase is going to bring CO2 in to PEP. It's going to be tough to draw out the whole pathway, but it's going to produce OAA, and then that's going to be malic acid, and then malic acid is going to go into the bundle sheath cells, as you can see here, bundle sheath cells, and then that is where the CO2 is going to be deposited, and that's where the Calvin cycle is going to occur. Rubisco is going to pick it up, sugar is going to be produced, and it's going to go into the vein, and then what's regenerated is, of course, PEP. And so it's a way to keep oxygen back, keep it back, and bring in CO2 so that carbon fixation can occur in the bundle sheath cells. It's rather clever. And so I find that to be very interesting. Now, this particular topic we had mentioned before, and so I'm going to go over it fairly quickly. This is talking about guard cells and how they're able to open and close and therefore mediate or regulate the amount of water that a plant uses and loses through transpiration. Now, there's a compromise here because ultimately transpiration is considered to be a good thing in moderation because, as you know, it moves water out of the leaf and therefore it actually causes the capillary action, the negative pressure, which generates the water potential necessary for water to move up a plant in general. But if it's opened too much, then you lose too much water and the plant's in big trouble. And so what happens is, in order to produce a situation where you're not losing so much water, some plants produce a lipid on the upper epidermis called the waxy cuticle. And that reduces water loss, and that's one thing. And so therefore, if you prevent water loss from up here, then you can control it down here. When you open the stomata, you could regulate water. You don't want it to be lost up here. Another function may be obvious, but I'll mention it. Another function of the epidermis is not only to protect from water loss, but it prevents the pathogens from getting in. And so the epidermis is a protective layer, and it also protects against water loss. And so these guard cells are rather important. And so the stomata allow gas exchange, oxygen out, carbon dioxide in, and also water out. And so they're the major sort of avenue for evaporative water loss of the plant called transpiration. It's a cool picture right here. You know what's interesting about this, in addition to the pulling effect that transpiration has, it also provides a cooling effect for the leaf. If you think about it, just like us, when we sweat, the warmest water molecules are the ones that are evaporating first, therefore giving a relative cool feeling. And so the same thing happens to the leaf. When water evaporates, it's the warmest water molecules that are leaving, therefore helps the plant stay cool in warm environment. If you notice here that during the light of the day, the stomata open up. And what's interesting is in the morning, when carbon dioxide comes in, let's see if we can do this. Carbon dioxide is going to come in like that, go inside the plant. Oxygen that's produced normally leaves. And also we have some water loss, and this is transpiration. And so to make food, the plant needs carbon dioxide to come in. And so this is the main avenue, guard cells opening and closing. So what happens is, here's the upper epidermis. This is a typical let's go here. This is a typical C3 plant. As you recall, upper epidermis, palisade, spongy mesophyll. Here's the guard cells down here. And this is where the gases circulate. What's interesting is not only is the structure of the lower spongy mesophyll interesting in terms of the spacing. The fact that the cells are sort of irregularly shaped and there's these little alcoves or pockets in here creates a real increase in surface area. And so this allows for the gases not only to move more easily, but it increases the surface area so that the carbon dioxide can reach all of the cells. I find that to be very important. So C4 plants are a lot more efficient because they can assimilate, as sort of a review, they can assimilate CO2 a lot more efficiently because they have the enzyme PEP carboxylase when the plant closes its stomata. And so it's sort of a trade-off. As you can see, for many species, it's sort of a ratio of 600 to 1, whereas in a C4 plant, it's 300 to 1. And so they're able to get more photosynthesis done with less transpirational loss when the stomata are partially closed because of that unique anatomy and biochemistry. And so what are the conditions that maximize transpirational loss? These might be obvious. And what's interesting, in the spring we're going to do an experiment looking at these conditions, but these are somewhat obvious. Obviously, in a sunny day where the sun's bearing down, that's going to increase transpirational losses. Obviously, when it's warm, that's going to increase transpirational losses. And when it's dry, likewise, more water is going to be leaving. And windy. Windy obviously causes, you think about it yourself, when it's windy, sometimes your lips become chapped as a result of the water evaporating from your skin. So these are the conditions that increase evaporation of water. Some plants have some really clever ways to reduce this. Some leaves are actually kind of furry and they have hair on the outside of their epidermis. And that's kind of cool because it reduces the wind across the surface and therefore it reduces transpirational losses. So basically, how do the guard cells open and close is our next question that we want to consider. And basically they open up in the morning because guard cells take in water through osmosis. And so in water, where does the water come from? It comes in from these other epidermal cells that are next to it. So water moves in to the guard cells and they swell open. And the reason they swell open is that there's these sort of radial bands of microfibers, but these microfibers are not protein, they're actually sugar of cellulose, and they sort of radiate out like spokes of a bicycle wheel. And what happens is when water moves in to these cells, let me see if I can illustrate that. When water moves into the cells begin to swell, and because these cellulose fibers are attached to the membrane, they cause a bowing to occur, a bending. And so the bending of the cell when they swell open because of the turgor pressure cause this to occur. This opens, and so this is how the CO2 comes in and the oxygen leaves. The question that we have is, why is water moving in? And the answer to that is they're moving in because the guard cells themselves are hypertonic. They have a high level of potassium, as it turns out. And this high level of potassium creates a hypertonic environment which pulls the water in. It lowers the water potential and water moves in simply through osmosis. And when the water goes in, they swell open. And in the evening, when they close, the water leaves and then they close up. So the question could be asked, okay, what's the story? How did potassium suddenly get in high concentration? So a close-up of this. How potassium became highly concentrated, we believe, is because the cell puts energy into this. And so what happens is, in the morning, the sunlight comes down, it strikes the chloroplasts, because remember, the guard cells have chloroplasts, and they're able to produce ATP. And as a result of that, it gets a little detailed, but that's fine. We can sort of handle this. We've earned this information. What happens is they use their ATP to actively pump hydrogen ions into the other epidermal cells. And by doing that, by pumping this out from low concentration to high, because it's now high out here, what happens is you've created sort of a membrane potential. It becomes positive out here, negative inside the cell. And as a result of that, an electrochemical gradient builds up, and so potassium ions, being that they're positive, let me just do this, get attracted to the inside of the cell because it's negative, and so those passively move into the cell. Interesting. And that's why it becomes very hypertonic, and then of course water then moves into the cells and they swell open. In the evening, when the sun goes down, there's no energy. The hydrogen ions then return. With no hydrogen ions, the potassium is not likely to go into the cell because it's neutral, and so therefore the potassium leaves. As the potassium leaves, the water leaves, and therefore the guard cells collapse. And so there you have it. And so this is a really cool picture of the stomata, and the guard cells look kind of pudgy right here. It really does look like a mouth. This is the scanning electron microscope here. And so as mentioned before in words, the cells use active transport to pump out protons, and that actually drives the potassium through. And so ATP ultimately is needed to open the stomata and close them. There are other, as I will mention in the future, there's other sort of hypotheses about how guard cells can open and close, and it's not related to potassium. It's related to circadian rhythm, and it's also related to concentration of carbon dioxide and temperature. We'll get into those later. Finally, there's the last topic in this chapter in photosynthesis is there's an elegant adaptation that some plants possess in the family Crassulaceae, up here, Crassulaceae. And what's interesting is these particular succulents, like ice plant or cacti, have a very fascinating way of photosynthesizing, because as you know, these particular plants tend to live in very, very dry environments. So this ice plant can grow right on sand, which is extremely dry because the water goes right through the sand, it percolates through, and so it's considered to be a desert-like habitat. And so what's interesting is, very right to the point, is these plants have an alternative photosynthesis pathway involving CO2 because they open their stomata at night. And I know what you're thinking, how could they do this? Because photosynthesis requires light. And if they open up their stomata at night, there's no point of it. But they're able to take in carbon dioxide at night. And so let me illustrate this. And so here's a cactus in the evening. And so carbon dioxide is taken in by the cactus or succulent, or if you will, cam plant at nighttime. The reason they do this is it's blazing hot during the day, and the plant would certainly die if it kept its stomata open. And what it does is the temperatures are usually lower at night, of course. And so what they do is they bring the carbon dioxide in at night, and they were able to fix it into other organic molecules in their mesophyll cells. And so they're able to store carbon dioxide in their vacuole, sort of like soda water. They're able to make bubbly water in the evening. And so at nighttime, they bring carbon dioxide in and they attach it to organic molecules that are already present in the chloroplast, like for example PEP. They were able to attach it, and this sort of looks familiar because it's similar to the C4 mechanism, but a little different. So it attaches it, it produces malic acid, and so if you think about this for a second, it's like this is how it's push the pen here. This is how it's hiding carbon dioxide in the plant. So in the vacuole. So this is malic acid in the vacuole. So this is kind of bubbly here in the vacuole. So carbon dioxide is hiding in the vacuole. And so then in the morning, they close their stomata, and then malic acid comes out of the vacuole, and it's converted into carbon dioxide and back into PEP. And so now the chloroplast has carbon dioxide, and it's ready to photosynthesize. And it doesn't lose a lot of transpiration during the day because it closes its stomata. And so I find that to be really cool. So in the evening, so this is at nighttime, the guard cells are open, CO2 comes in from the cactus, it's able to incorporate it in the vacuole into malic acid, and then in the morning, when they close their stomata, because it's getting really warm, the carbon dioxide is released, and therefore they're able to photosynthesize during the day. Because during the day, that's when ATP and NADPH is being produced in a light reaction. And so, examples of a C4 plant, in addition to sugarcane, in addition to corn, there's some other grasses, like for example, crabgrass. This is something that if you have a garden, you're very upset at, because this crabgrass tends to grow very, very quickly. It's very invasive, and interesting, it's a C4, so it's able to do really well under dry conditions, and so that can be a problem if you want it. So corn is not a problem, but crabgrass is a problem. And then pineapple is a cool example as well, because pineapple is capable of being a can plant. And so pineapple can, again, sort of thrive in warm environment. And so it can open up its stomata at night and fix CO2, and then during the day produce glucose, and then C4 plants are able to do this similarly, but with a different sort of pathway. So in conclusion, the light reaction occurs during the day, so does the dark reaction. It also occurs during the day. Why? Because you need ATP and NADPH. And so this Calvin cycle, one of the great pathways in photosynthesis and one of the great pathways in biology, ultimately produces glucose, then potentially sucrose and starch. And it is really the input of all carbon, all organic carbon, to produce proteins and oils and nucleic acids. So I hope you enjoyed that discussion of photosynthesis, in particular the Calvin cycle, photorespiration, and can plant photosynthesis. Thanks for watching.